Topic is antibodies. So, antibodies are immunoglobulin Ig protein molecules synthesized on exposure to antigen that can combine specifically with the antigen. So, the antibodies are immunoglobulin, and they are synthesized when the pathogen, that is the antigen, is exposed in our body. That time antibodies are synthesized. So whenever pathogen enter our body, the B lymphocytes produces an that is army of proteins called antibodies to fight with them. So whenever the pathogen enter in our body, that time the B cells produces the antibodies in large amount to fight with the pathogen. So thus they are secreted in response to an antigen by the effect of B cells called plasma cells. So how they are produced against the antigen? So and they are produced in the B cells which are called as plasma cells. So the antibodies are classified into 5 major types based on the physiological and biochemical properties. Ig, that is immunoglobulin, G means gamma. IgM, that is immunoglobulin mu. IgA, immunoglobulin alpha. IgD, immunoglobulin delta. IgE, immunoglobulin epsilon. So in 1950, Porter and Edelman had done an experiment on the basic structure of immunoglobulin. It is in the letter shape Y. And you can see the antigen binding site. And this site, this is the antigen binding site. Opposite to this site, it is the variable region site. And you can see a light chain, that is you have two light chain, this one and this one. And these are connected with, that is the light chain and the heavy chain. This is the heavy chain, all in two, that is the four in structure. So this one is light chain, that is this one and this one, it is the light chain, and this one is the heavy chain. They are binded with the disulfide bond. And you can see the constant region also. This is the structure of immunoglobulin. This they can ask in your examination to draw the structure of immunoglobulin or describe the structure of immunoglobulin. So first, that is, they have done an experiment to know the structure of immunoglobulin. According to the experiment, they get the antibody model, that is Y-shaped in structure, and it is made up of 4 polypeptide chains. So this is made up of 4 polypeptide chain, that is 2 is light chain and 2 is heavy chain, which is denoted as H and L. So L denotes the light chain, which is identical. 2 identical means 2 same light chain, and they have the molecular weight of 25,000 Daltons. Dalton is the unit of molecular weight, and it comprises 214, that is 214 amino acids in the light chain. And you have same way the 2 identical heavy chains. It is denoted by H, and its molecular formula is 50,000 Daltons. So which has approximately 450 amino acids. And these chains are linked by the disulfide bonds. So this is the disulfide bonds which links the H and L chain. Okay. So one light chain is attached to the each heavy chain, and 2 heavy chains are attached to each other to form the Y structure, that is Y structure. You can see this one, that is this side light chain was attached to heavy chain, and the heavy chain is again attached to the light chain, 2 sides, and in the middle the 2 heavy chains are also attached with the disulfide bonds, and due to this only you are getting the letter Y shaped model, Y shaped structure. Okay, Y shaped structure. So an antibody has H2L2, that is So this one L, this one, so 2, and this 2 are heavy. So that the antibody has H2 and L2. So the heavy chains are flexible hinge region, that is in the approximate middle. So in the middle region they have flexible one. So they are flexible. So each chain has two terminals. So this side one terminal, this one terminal, and this one terminal. Okay. So they have terminals, two terminals. Each one has two terminals. This one and this is also one terminal. Okay. So for L chain, and H chain, this one terminal, this one terminal, and this one terminal, this one terminal, and this one terminal, like that. Okay. So they have variable V region at one end and a much larger constant C region at another end. So you can see here, this one end you have the variable region, and here the large one, this is called the constant region, which is called as C region. So variable region with V and constant region is C. So the antibody responding to different antigen have very different variable region. So according to the antigen, the variable region has differences, but the C region will be same for all the antibodies. So there is no change in the C region. In each arm of the monomer antibody, the variable region of heavy and light chains combine to form an antigen bonding site. That is, in this variable region only, these two L and H combine to form the antigen binding site. There only, this region only, the antigen will be bind with the antibody, and the antibody fight and kills the antigen. Okay, so that is called fit shaped. That is the shape, the binding site, no, it forms the fit shaped. Fit as a specific antigen determined. So that particular portion is called as the fit. Okay, so each antibody monomer has two such antigen binding regions. So each one, because it is white, no, this one antigen and this one. So you have two antigen binding site. Okay. Then the C region that forms the stem of the antibody monomer determine the antibody class and serve the common function in all antibodies. So this is the stem region. It is constant. It finds the antibodies of what type it is, and it is same for all the antibodies. Okay. Functions of immunoglobulin. Agglutinin. If an antibody, that is antigen, was entering into an body, in our body, first the antibody, what will this immunoglobulin do means, it will agglutinize. So it will agglutinize means it creates a cover and it freezes the antigen. Precipitation. So during agglutinization itself, you can precipitate the antigen. Okay, that is one process. After precipitation and agglutinization over, you can neutralize, that is, you can minimize the poisonous activity of the antigen, and you can able to neutralize also. This function was done by the immunoglobulin. So the immunoglobulin functions are agglutinization, precipitation, and neutralization. This they can ask in 2 marks as well as 3 marks. Hope you understand the topic. If you have any doubts, post the doubts in the comment box. If you want to watch the video in Tamil, I had given the link in the description box. You can go and watch it. Give a thumbs up, share and subscribe to Science Easy Tech channel.